Hello, my colleagues and sincere teacher. In this part of our term project presentation, we will discuss some points about what is lymphatic system, how it works, how lymphoid cells differentiate and diverge, and types of lymphomas and their symptoms. In addition to that, while we enlighten you with the theoretical background of our project, we will also shortly mention about studies related with lymphomas and neutrophils. Now I am letting my colleagues to talk, and I recommend you to sit back and enjoy.
Lymphatic system is a part of circulatory system and immune system. It is network of tissue and organs that help to get rid of body toxins, water, and other unwanted materials. The primary function of lymphatic system is to transport lymph, a fluid containing infection-fighting white blood cells throughout the body. Unlike cardiovascular system, it is not a closed system. The lymphatic system primarily consists of lymphatic vessels, which are similar to the circulatory system veins and capillaries. The vessels are connected to lymph nodes where the lymph is filtered. The tonsils, adenoids, spleen, and thymus are all part of the lymphatic system. Lymph is a clear and colorless fluid. The word lymph comes from the Latin word lymphia, which means connected to water, according to the national lymphedema network. Unlike blood, which flows throughout the body in continual loop, lymph flows only one direction. There are hundreds of lymph nodes in human body. They are located deep inside the body, such as around the lungs, heart, or closer to the surface, such as under the arm or groin, according to the American Cancer Society. The thymus located in the chest above the heart, according to the Merck manual. This small organ stores immature lymphocytes and prepares them to become active T cells, which help destroy infected and cancerous cells. Tonsils are large clusters of lymphatic cells found in pharynx, a first line of defense as part of the immune system, according to the American of Otolaryngology. The most common disease of the lymphatic system are enlargement of the lymph nodes, swelling due to lymph nodes, blockage, and cancers involving the lymphatic systems, according to Dr. James Hamrick. They are called leukemia when in the blood or marrow, and lymphomia when in the lymphatic tissue. They are grouped together under the name of lymphoid malignancy.
All in all, we may summarize aspects that our project covered in order as follows. Neutrophils are the most common white blood cells type. They are the first migrated cell when an inflammation occurs in body. They are the main points of the innate immune system. And because of being the main points of the innate immune system, they have a big role in cancer biology. Neutrophils are associated studies which are worked on the malignant transformation, tumor progressing, angiogenesis, and modulation of the anti-tumor immunity.
Since this image demonstrates that tumor microenvironments have a significant effect on hematopoietic stem cells differentiation, also myeloid-derived suppressive cells may happen to be generated at the end of the development. As explained and described in detail in the next section, through successively conducted experiments have shown macrophages' effects on tumor angiogenesis, metastasis, and also improving tumor solidity and malignancy. Here in this depiction of myeloid precursors, Differentiation, mostly regarded as strikeful, lead to show innate immune system parts, myeloid-originated cells divergence, which in particularly be dwelled on granulocytic path. Basically, this granulocytic path that followed give rise of polymorphonuclear cells, basophils, eosinophils, and our key player, neutrophils. Indeed, each of innate immune system cells, such as mast cells, dendritic cells, and these granulocytes, have intrinsic roles and responses against any external stimuli, may happen to be as a skin cuts, foreign matters, or any microbial invasion. However, due to our subject, we are mostly into neutrophils and their correspondences depending on any eminent occasion. Innate immune system mostly overwhelmed by neutrophil abundancy, and they take place in almost every inflammatory responses. Fundamentally, their job is to be recruited upon where the signals come from and clear all cellular debris, intra or extra cellular debris contained in this. Secreting cytokines and other varying proteins to attract lymphoid-originated cells to proceed. These cells, B lymphocytes, helper T cells, etc., also associates with antibody release and aberrant cells perishing, harvesting. However, after that response, neutrophils swell and cleanse whatever left remained. These includes antibodies and or destroyed cell particles and many more. Cellular signals, secondary signal materials and chemicals are included in this mass for sure, which also another discussion case for the further part of this video. Innate immune cells, which includes mast cells, natural-born killer cells, granulocytes, and many more types of them, are consisting the very first defense line of the body. In any severe or trivial deformation of tissue, any foreign microorganism invasion or external stimuli that damages to the skin counteracted by innate immune cells and probable inflammatory responses as a consequence. Recent studies about innate immunity associated with tumorigenesis, especially promotion and prognosis, state that tumor microenvironment changes and malignancy of tumors probably depends on these cells' communication interaction with tumors' microenvironments. Moreover, according to accomplished other studies, chronic immune responses have been compromised as being critical for tumor progression, metastasis, and chemotherapeutic agent sensitivity over tumor cells. Ever since macrophages' role in regulation of tumor cell responses have a great impact on tumor anti-cancer treatment sensitivity, yet neutrophil cells are not known whether be effective on anti-cancer cell treatment sensitivity or not. In this picture, you may easily see tumor development associated with inflammatory responses and inflammation. In this depiction, neutrophils and most of innate immune cells secreted signaling molecules such as cytokines, as demonstrated in the very bottom of the picture, and their association and impact on tumorigenic development and malignancy of tumor cells. In addition to all, you may easily see its impact on metastatic cells generation and also immune cells surveillance around the inflammatory responses occurred. Corresponding to this, main goals of our project were predetermined followingly. Utilization of faster and more credible method than native strategies and used old school techniques until nowadays to separate neutrophils from chronic leukemic lymphoma patients' blood samples. As a second goal of our project, differentiation of HL60 cells, which are determined as pro-myelolytic cell lines, into neutrophil-like cells through granulocytic pathway in order to study their role in alteration of sensitivity of lymphoma cells against anti-tumor cancer treatments, therapeutic agents, and chemicals. Microorganisms invasion possess more prominent role, which is rise effective innate immunity responses. Besides those described, some variable effector functions in several pathological conditions can be considered as their responsibility, mainly inflammatory responses and inflammation and inflammatory responses. Therefore, in vivo studies that solely requires neutrophil isolation can be utilized whilst they spare times and kept low in their expenditures, facilitate execution of researches associated with neutrophil mediated immunological functioning in vivo and ex vivo studies.
Acquiring neutrophils in high purity and yield is possible by using density gradient separation methods. The other methods for isolation neutrophils are not credible as much as density gradient separation and the other methods like neutrophil isolation kits. Neutrophil isolation kits are not trustworthy since they use specific antibodies for specific neutrophils. These antibodies may cause activation or differentiation of neutrophils with the interaction with each other. But density gradient method not causing this problem and allows us to separate pure cells with functional integrity. Another advantage point of gradient separation method over neutrophil isolation kit is that kits are much more expensive than the gradient separation method. If you take a closer look, overall working principle of density gradient separation technique for our project, it contains gradient solution in the bottom and of the tube and diluted human blood followed by the centrifugation at 300 G for 35 minutes in non-stop fashion. At the end of this procedure, we expected to see mononuclear cells at the interface and neutrophil residues below the former. So this method elaborates neutrophils with high yield definition. In order to sustain tumogenic environment, 3D cultivation performed in the next phase of the experiment. So, since neutrophils has a short life for in vitro experiments, usage of fresh human neutrophils is not an option. Instead of that, premyelocytic HL60 cells are used via the usage of differentiation inducers such as dimethyl sulfate, DMSO, and retionic acid. And creation of neutrophil-like cells can be acquired in this way. So that sorting will manage to prevent of having different responses of neutrophils due to the isolation of different donors. In vitro 3D culture methods has a great advantage over 2D culture methods. While 2D culture methods uses spread on plastic surface and provide unnatural cell attachments, 3D culture methods enables us to provide cell-to-cell interactions. So, to be specific, 3D culturing enables us to have physiological environment. Thanks to these beneficial features of 3D culturing, 3D co-culture methods can be used, especially in cancer cells and other cell types also, to evaluate their contribution of tumor growth, angiogenesis and metastasis. Today we have talked about our introduction part of our experiment. We try to clarify some decent points about background, theoretical background of our experiment. So next week we will going to talk about our protocols, and we will try to clarify some points in the protocol that will help us to achieve this experiment. Thanks for listening.